been a long time since we've done a Biota recording with Bruce Damer. You have previously been a punctuation in the Biota recordings. I'm sorry, I need to call you Dr. Bruce Damer now. My wife and I have done the spiritual trek down Bear Creek Road, and we are at the farm here with Bruce. Bruce, I know you have a current book project, but for folks who maybe just caught up associated with the ChemoGrid, can you take us from the ChemoGrid to where you are currently, or the start of the ChemoGrid in terms of assembling all the plastic bits and pieces to where you are currently? What has gone on in the past, I don't know, year or so?
Well, we completed all the EvoGrid simulations, and the EvoGrid was an all-computer, complete silicon-based, if you say almost like an emergence system, where we use GROMACS, molecular dynamics, like thousands or hundreds of thousands of those instances, small volume chemical simulations running, and then search and selection to hill climb through chemical space to get the highest performing experiment. Had 189 molecules formed out of 1,000 atoms, and they were all different types of organic molecules, pretty much. And so that was experiment number 6, and it occurred on April 15th of 2011, where we saw the staircasing. So the magic formula, or the magic sort of journey pathway to find, is a staircasing effect, where you have a bunch of complex molecules forming in, say, hundreds of experiments. They can't move it to any higher maxima, so you pick randomly from them, wiggle the parameters, and generate hundreds of new experiments, and then see if they manage to get to a higher maximum. And by using a technique called stochastic hill climbing, we found the doorway through. And so literally by the end of experiment number 6, which ran on 30 cores for 2.5, 3 months, we had this 189 molecular product. We had sequestered about half of the atomic population into molecules. So that was good enough to defend a PhD with.
So a question that I got at the A-Life conference, and apologies that I can't remember which of the many folk who came up to me associated with these recordings, but someone did ask me, if the EvoGrid was to continue, if we were to take exactly what you did and just kind of progress on with it, with potentially more cores, potentially faster computing, potentially even removing the chemistry simulation, putting a new chemistry simulation in, do you think that you would see a progression, maybe not as steep as you initially saw, but do you see that there was enough in the EvoGrid that if there was a devoted team that was looking at it continuing on, that it would get more results, more productive results, basically, in the long term?
I concluded, I was actually sitting at the Origins 2011 conference, which was sort of the Origin of Life mega conference in Montpellier, France. I was sitting on a park bench, depressed, because I had worked out in my head that we were 4 orders of magnitude short of the computing power needed to do even basic Origin of Life experiments in terms of the volume you need to simulate and the number of molecular products and the time frames.
But the argument was made, and certainly I'm sympathetic to this, that kind of computing is probably 5 to 10 years away, and if you look at the nature of GROMACS and replace GROMACS with something that's considerably more optimized for an EvoGrid-like space, then that comes down in time as well. I mean, I think the computer ceiling that you would need to see, the computational ceiling that you would need to be 10 to 15 orders of magnitude to eliminate a human lifetime associated with just the progressive computing that we've seen. And I think certainly my own discussion and the questions that came to me through the Artificial Life Conference related to replacing GROMACS in particular, but also looking at vastly more distribution in the EvoGrid model. I think there was certainly a narrative which was recorded through these podcasts associated with the hope of the EvoGrid. And I think as this audio went out to tens, hundreds of people, it stuck with the number in the community that they still, irrespective of your doubts, have a sense that this is still a potential way. And I think in terms of the kind of meta-EvoGrid, the notion of this framework that has an internal chemistry engine, the state reproducing, multiple potential future states, all these kind of things. The blueprint of the EvoGrid still is relatively robust for expanding computation. Mm-hmm. And I think the general sense, I mean, although maybe I'm kind of getting the community as a kind of focal point associated with this topic, but I think there seemed to be an underlying sense that the computation still held some potential. And I think particularly your discussion at Contact, and unfortunately we don't have the day following audio that you recorded with Randall Hayes, but I think there are still some optimists out there in the community associated with this model. To those folk, what would you like to say?
I sat on this park bench with another researcher, and it occurred to me, as I said, in my lifetime, I'm not going to see computers crack the origin of life. Emergent phenomena molecularly modeled so they can be reproduced in the lab. And then it occurred to me, replace the digital simulation of chemistry with chemistry. And I actually had this strange vision of inflating party balloons where you could then squirt a sample, experimental solution, use the positive pressure of the balloon to actually then resample back out and do thousands, hundreds, thousands, millions of small chemical experiments. And then in a flash, it was in my head I said, wait a minute, if the molecules simulate themselves and you have a parallel computing engine running that is using artificial life techniques, it's using basically genetic algorithms because it will watch these experiments in a vision system or a mass spec or a combination of things and grade them and weight them and then restart new experiments. You could flush the system out and start 1,000 new chemical. You let the chemicals simulate themselves.
So that's the ChemoGrid. And in addition, there were a number of questions associated with the ChemoGrid. But returning just perhaps slightly to the EvoGrid idea, for folks who are interested in kind of continuing on with the EvoGrid that maybe are not as pessimistic as you are.
I would say to them, use your idea of chemical automata, because basically using a dissipative dynamic system like GROMACS, trying to model real molecular dynamics, it's dissipative dynamics. So the vast majority of things that are going on are molecule-to-molecule contacts that don't do anything. Mm-hmm. And that's the frustration of computer science people, that to simulate nature, you need to simulate a vast amount of no-ops, basically. Mm-hmm. Random encounters, heat moving through the system, etc., etc. So very little happens, like Will Wright said, very little happens for a very long period of time. But if you want to use the EvoGrid's search and selection hill climbing technique, do it with chemical automata where interactions have a higher meaning, where it's almost like the Wolfram approach, where an encounter between two things does produce something.
So the idea of event-based versus time-based.
Exactly.
So you take the event scale rather than the time scale and actually look at the event interaction.
So for example, here'd be a very good, if you could use chemical automata that have high meaning in their interaction, in their events, to simulate the action of lipids. Lipid chemistry is sort of, I think, where it's happening. Having hung out with Dave Deemer for a couple of years now, I'm a convert to lipid chemistry, but the behavior of lipid is so fascinating. It's so freaky. I mean, people have to understand, your cells are made of lipids, phospholipids. They basically nestle together with their heads and tails, the tails together, the heads facing outward, and they ripple and they flip. They flip in orientation and they let things through the membrane. They're not chemically bonded. This is an affinity, a molecular affinity. So membranes have permeability, and it's permeability that is adjustable. The membrane can also grow by adding more lipid. It's one of the weirdest phenomena in the universe, and it's so weird that it can support the emergence of a living system. And it's for free. This is not a biological system. This is a natural system. And the Earth was populated by lipid from incoming meteorites. This is what Dave Deamer showed in the 80s when they sampled the Murchison meteorite and put it in solution. There were lipid membranes forming. So lipid chemistry can be modeled with computers. You don't have to model every single atom and its behavior. You can model all kinds of vesicle formation, all kinds of permeability. Also, you can model polymerases, polymer chemistry, because you don't have to do everything. You just model the entire polymer, and things like replicases. So all of that could be done extremely effectively with a traditional all-silicon EvoGrid. And then what you do is you marry that model to a real ChemoGrid and say, we've observed the formation of polymers inside vesicles that then regulate the growth of the vesicle, which is an important result in simulation. Can we now build the chemical experiment and compare it to our model all the way up and find out how far off we are?
So let's talk about the ChemoGrid, with the view that the folks listening in have heard the description that you've given, basically, in our prior recording and also in this recording. One of the interesting things that happened, and thankfully it was recorded and put out in a podcast, was at my Artificial Life in Industry talk, because we had wet artificial life folk there. And I talked a little bit about the ChemoGrid in terms of the hobbyist wet artificial life community. And initially, and this was perfectly captured in the audio, there was a little bit of skepticism. But then I explained the kind of broader principles of the ChemoGrid very much in traditional artificial life terms, associated with how the experiments would go and how things would be selected and how it would progress accordingly. And then I think they began to understand that this was a kind of hobbyist wet artificial life. So in terms of your own thinking of the ChemoGrid, can you frame it in wet artificial life terms?
If you could, and there's a high school science teacher building, he's the first person he's writing for the book, and he's building a ChemoGrid Genesis engine experiment in his basement in Illinois. But what he's doing is he's saying it's really, you want to model an analog of the real world. So based on what Dave Deamer's guided him, he said take a sample of volcanic rock, drill a hole in it, have hot water squirt up through that, but also come into contact with the bits of the volcanic rock, spray out onto little trays that are moving. And on those little trays are other rock samples. They move around, they click around, there could be 50 of them or whatever number. They dry, but then they get re-impugned with wet, hot rock, which has also come through this volcanic pipe. And then what you do is after minutes, hours, or days, you scrape off those samples and you put it under, or if you're really good, you've got a robotic system to automatically look at them, and you see how many amino acid chains have formed, how much lipid chemistry has occurred, and you're modeling the geyser environments that are they're a real potential for the early origin of life.
So in terms of contamination, because I think this was my concern when we first talked, and I tried to explain this at the Artificial Life in Industry discussion as well, associated with, I think you used the term, use some large British ship to represent the chemistry, the organics that we see, and looking underneath that, so really eliminating the top noise band and perhaps the bottom noise band and looking at a kind of nutritious central layer. But can you talk a little bit about contamination in the system, and if you have, for example, if you're doing this, I've been into some basements in Illinois in recent times, and the quality of the organics in those kind of environments, just at the air alone, is pretty good.
Dave actually believes ordinary household bleach will really knock out pretty much.
Wow.
And heating. The water, and of course there are extremophiles, but the bleaching of the environment will really, really knock out. And you use an ordinary enclosure like an aquarium, a sealed aquarium. And he believes that what you're looking for anyway clearly if you saw a bacterium that looks like the complexity of the Queen Mary compared to a rubber ducky, you would just discount that. And I was also asking, what about, leftover biological stuff? And you can't, nobody's going to really make an analog of the early Earth because we just don't know the atmospheric content. We don't know anything. We know a little, or we can make guesses. But so his ChemoGrid is very much, the mineral surfaces, etc., but there are ways to do chemogrids with microfluidics. You can do it with a glassware. We built a system last summer that was glassware, and it was we built lucite panels with really simple, small test tubes. So the idea is, in some of these chemogrids, is closed loop. So you have the test tube, or you even have a beaker flask, and you put a stopper in the end, and the stopper has 2 ports in it, and you put, basically you snap off pipettes, shove them in there. It actually creates a pretty good seal, and then you put your glass tube, your plastic tubing through, and you can pressurize the system. You can rock the experiment back and forth. You can heat it from below. You can do all these really, you can use an Arduino to control a whole shelf of these things that are rocking back and forth. Some of them are in darkness, some of them are, etc. And then when you want to sample, what you do is you have a stopcock that you could turn automatically or by hand. And as soon as you turn it, it sucks basically a bunch of the fluid out of one of these test tubes down into an observing area where you could actually look at vesicles that were formed, and then you turn the stopcock, you turn the pool pump, the aquarium pump the other way, and it will pull the sample back in, completely pull it back in, so you don't denude your samples.
But in terms of the pump is going to be your greatest contaminant in that kind of environment.
Well, as long as the air, it's a closed system, so there's a fixed amount of air in it, and the pump is basically squeezing a diaphragm of existing air. So it's completely closed.
Oh, okay. So the pump doesn't introduce anything into it.
Doesn't introduce anything. Okay. It's just squeezing this diaphragm. So for cheap, this is maker faire kind of. For cheap, you can even do this with glassware. Microfluidics is the next step where this is what Steen and Rasmussen and the lab wants to do with their life cycle. Microfluidics is basically, taking silicon chip technology and making thousands, and in medical testing they're doing these things for you can pinprick your skin to do a blood test, and connected to this prick is a chip that is going to do the blood test, and it's got a USB port on it, and you plug it into the computer, and it's already done the test. It's cholesterol, PSA test or something. So there's tiny labs on a chip. There's a third. There's a next method is the 96 well plate technique, and this is from Affymetrix, and this is from the Human Genome Project, where you have an aluminum plate or a glass plate with small wells carved in it, and such a system has been built at NASA Ames for astrobiology. It was built at UC Santa Cruz, and it's being operated. And they literally rotate this plate around and dribble individual little wells full of, in this case it's a lipid polymerase experiment, and then they dry them out with a drying station, and then the little wells continue along, and they're rehydrated, and then they go. And so that's another Genesis engine. That's another ChemoGrid. There's a fourth model, and this comes from Dave Deemer again. The fourth model for the ChemoGrid is using nature's own containers. So instead of glassware, well plates, or little channels and silicon chips, what you can do is, if you start in the morning with egg yolk, you can do a series of processes that will pull out, basically a little pile of powder, white powder, which is phospholipid, out of that egg yolk. And then if you take a pinch of that, you're talking a gram, throw that into a solution, into water, it will form billions of small containers, lipid containers. There's so much lipid. So what you do is you say, all right, well, I formed 1 trillion containers, some of which may have contents in them, some of which don't. What I can do is dry that whole system out, the containers flatten out and become a multi-lamellar, multi-layered structure. They go from 3D, and as the water leaves the system, it goes down to 2D, concentrates all the material. The water's gone out, so it doesn't support back reactions. And then you find long-chain polymers form, and all kinds of things form. You put a drop of water back, and those trillion vesicles reinfuse, and now they have complex molecular products in them. So you're doing a trillion experiments in one shot. That's big numbers. And that's probably the numbers that you need. And then what you would do is take those reinfused vesicles and flow them through some kind of experimental environment where, and this goes back to Irene Chen's paper about 5 years ago. If you could create, an untold number of vesicles with random contents, this is Freeman Dyson's dirty garbage bags of dirty water idea, then what you do is you run them through a test. You run them through an environment where they pop or don't pop. That's a form of selection. Even though they're not living systems, they're still growing because lipid are still in the environment and they're still growing and they're elongating and they'll butt apart. In fact, Jack Shostak has recently at Harvard or Massachusetts General Hospital, where their lab is, they've observed these vesicles that have formed getting longer and longer. They're like party balloons that people blow up and make dogs.
Yes.
They will break up all at once in an effect called purling, where there's a chain of vesicles formed. Like, it's not just one division, it's multiple divisions. So lipid chemistry rocks in terms of producing containers with contents of different sizes. So I'm convinced that the whole target for Genesis engines should be the creation of trillions and trillions and trillions of lipid vesicles with different contents. And then what you do is observe those, observe a zoo of lipid vesicles, put a certain selection pressure on them, and see which ones come through. So you can basically dye them. You can see the contents. You can do a mass spec on them, but then you have to destroy them to see what the contents are. But they're big enough that with an electron microscope, you should be able to characterize them. So they're going past the microscope. You get a digital image. You find that experiment number 1,500,000,000 such and such is producing a lot of identical sized or almost identically sized vesicles that seem to have similar contents. You've got a marker. So then you start 1,000 more of those with slightly different parameters and see if you get even more identically sized ones. And you find out you have a chemical species of vesicle that just forms with these conditions. And that species, it's not a biological species, it's a chemical structure that persists. And then the whole game is, how did they persist? How did they keep growing and divide? And how did they get the same contents on either side of the division? And then you would go back to Freeman's idea of catalysis happening statistically, which goes back to Stuart Kaufman's ideas of that you need an autocatalytic network.
Certainly.
So then the whole thing just emerges because now you've got a statistically reproducing, contents of the egg. You've got the shell of the egg and the division happening. So now there's two eggs, and you've got statistically the same, similar contents. And if those contents persist long enough and allow the vesicle to keep growing and then divide again, you have a total indicator on the way to a chemically alive system. So then natural selection has to kick in. And if you have natural selection, those chemical contents and the mechanism that reproduces them for both sides of the daughter cells is now being selected for. So the machinery is getting better and better. And that would be if the Genesis engine showed an eternal bubbles that never popped, where you had hundreds of lines of division going on of these bubbles basically, and even if you couldn't detect the machinery that was doing it, would be a major breakthrough in science, because you have allowed a chemical system to evolve before it became a living system, the machinery to persist a mega molecular structure and through multiple divisions. That's a huge result.
So in terms of folk who are currently assembling the chemistry sets to create the ChemoGrid, this description has a kind of continuum from the ChemoGrid to Dina's work. But in terms of the folk that are actually looking to start this, in terms of putting together the basic vessels, potentially starting to experiment with dried egg and these kind of things, as a blueprint for them, as a kind of hobbyist blueprint, very similar to the hobbyist robotics community, do you see that there'll be a couple of companies that create their own dried egg plastic vessels that they send off in kits for $25? Or how do you see the hobbyist element reaching towards this goal?
I think the hobbyists should use ordinary test tubes, ordinary cheapo glassware you can sterilize, that you can get in any science shop or any throwaway stuff from high school labs. But think about, if you say you've got a lot of very small test tubes with stoppers in them, and you're putting different solutions, think about how you build arrays, grids, shelf systems that do different things, that will heat one sample and not heat the other, that will agitate a sample, where you can put a small piece of mineral in them, because that was the early Earth. It was a mineral and water environment that then you can somehow automatically look at. The whole thing is automation, because if we can't do automation, we'll never get anywhere. I mean, that's one of the problems in origin of life research. It's handwork. And they're doing one experiment that takes months or even years to set up, and then they often miss what went on. One of the labs I went to, I said, well, how? You've got all these, you have a tray of test tubes that have been treated differently, and you take them out day after day, and you're putting them into this $500,000 microscope, and you're looking by your own eye. How do you know you? And they said, we don't know. We could have missed the whole story.
Yes.
It could have been happening in this test tube here.
400 times over.
400 times over, we completely missed it. So there's a huge error rate. And so the idea is what we built here, here at this house, was little camshafts that would tilt our test tube. And you can think of how doing that. You don't even need a shelf. You just have the test tube with rubber bands, and the little camshaft push the test tube so that the solution was against one of the stoppers. The stopcock turned, pulled the sample so that you can look at it. Now, ideally, what you would do is, of course, look directly into the test tube, but then you've got to have your microscope somehow moving along an assembly line. So you either sample out of these hundreds of tubes into a common, imaging system, or you have to be able to look through into the sample. So either way, what Dave Deemer's team has done is they're moving the samples around on this plate, on this 96 well plate thing. So then you have to decide what it is you're looking for. What part of the experiment are you going to look at? This high school science teacher, he's looking for polymerization on hot rocks that dry out.
And so in terms of, this is another question, in terms of temperature, in terms of tilt, in terms of light, in terms of a wide variety of factors, what would your top 4 or 5 list be of variables that people should look to make constant, potentially, and vary one of the variables, or potentially vary multiple variables? What do you think the variables are for the hobbyist level ChemoGrid?
There's two things. It's really the content of the experiment. So the amount of lipid is there, the amount of, you might even put nucleotides of some sort, or amino acids, things like that. The concentration, salinity matters a lot, pH matters a lot. So
if you've bleached it already, you're already in very difficult conditions associated with some of these factors. So maybe lighten on the bleach for certain experiments, or potentially remove the bleach?
You would remove the bleach. You basically would bleach out your gear, let it dry. Let it dry in a as close to a sanitized environment as you can, not open to the air, because there's all kinds of things floating in the air. Will just find their way in. or just washing out with bleach and then washing out with water that's decontaminated as well. So because you're going to end up having water in there anyway. So then you end up with deciding, and you can titrate in. You could have a system by which you don't have to do this by hand. I mean, there are automated systems that will titrate in, just set the pH and set. They're fairly cheap. I mean, high school groups use this. But then what matters? It may matter that there are mineral surfaces. It's not just. So you decide what rock samples matter. But also what matters is temperature. Temperature. So there's an idea that temperature fluctuation matters because you need reaction energies to make these reactions happen. Otherwise, it won't happen in your lifetime. It's probabilistic. Evaporation could matter a lot. But also, maybe that agitation matters or it doesn't matter. Stirring. You could have a magnetic stirrer that goes on every once in a while. Or you could move the sample over top of a piece of mineral and have drying occur. And then, so it's really, there's hundreds of thousands of variations on this. And as a hobbyist, you would decide, I like the idea of simulating the volcanic shoreline. Or I like the idea of the hydrothermal vent. So mine are going to be hot. There's going to be flows, not only past mineral surfaces, but this is the work being done at JPL, where they build analogs to deep hydrothermal vents. And they find that in these vents, the mineral waters almost are going through little worm-like cavities the contention by the JPL crew is these are perfect RNA making machines that are at these hydrothermal vents. So you could say, I'm going to do that, but I'll do it with, a little hose, almost like a heat exchanger that you're going to build, and see that by the end of the heat exchanger, you've got a concentrated RNA. So you could, what the approach of the Genesis engine or the ChemoGrid is that a lot of independent experiments could be done and then combined. So if you were an RNA generating, long chain RNA generating system, you would then attach that to somebody who's doing lipid chemistry at the shoreline and say, let's have the outputs from both combine. So now the RNA is appearing in a wet drying cycle. And that would be analogous to a shoreline, like a black smoker, like a geothermal vent.
Certainly.
That's at the shore that is spewing out RNA product. There's wave action going over it and hitting the shoreline, and those waves are full of lipids. And all this slurry is on the shoreline, and it's now wet drying. And you might find that, what chemists call amplification, that this amplifies, vastly amplifies the formation of these polymers, important polymers, inside of containers. And you would be able to write a paper in a major journal that would say, hey, if you just combine these two relatively simple techniques, analogs, to the real world, you get vast amplification of this. So is this plausible in nature? Probably. Here's the environments, the two environments we're combining. So it's a collaboration. Then you might find, 1,000 nmer RNA chains that could become replicases inside vesicles. That's non-trivial. And most of these labs out there usually only do one experiment. They don't try to combine and make whole complex environments.
Yes.
And then the addition of mineral where they're drying, then you swap out the mineral surfaces and say, well, volcanic, what about clays? How do clays encourage? And there's a lot of theory behind how clay encourages the formation of short chain organic molecules. So somebody swaps clay out in their ChemoGrid, and then they amplify it even more. It's a tremendous, these things will take years, but what you can do is if your ChemoGrid has generated this really cool result, it's publishing its results always to the net. Somebody else could take those results and automatically run their ChemoGrid apparatus in their basement, take your experiment further, which was always the dream with artificial life, that you build a common...
Yes, it's a non-trivial problem, though, and I think that there's a podcast in and of that problem. In fact, we've recorded a couple previously associated with artificial life. But in terms of time, I did want to get to the book, and I know we can expand all of this in greater detail with future conversations. But the book, specifically as it is now, for folks listening in, can you introduce what this new book is and what it's going to achieve?
Well, in a sense, writing the PhD dissertation, which is sitting right next to Tom here, I was depressed on the park bench, had this insight about using chemicals to simulate themselves, roared off to Dublin, defended the PhD, not feeling as though it was a complete failure, because the higher goal was there. The PhD material worked, and we found this magic novelty, enhancing technique, accelerating technique. So then a week after that, after defending the PhD, and it might have been a day or two after that, I wrote to Joseph Seckbach and said, I think there's enough here for a book. And it's not only a book, but it's an attempt to frame a new field or subfield, an approach to origin of life that hasn't been taken yet, which is massive scale industrialization of these experiments. We're not going to get anywhere unless we can massively scale up the system with the best computing, with the best knowledge of how to run experiments and how to hill climb and how to sample and how to use basically genetic algorithms to drive the hill climb faster and faster, to use the best teleological concepts, the best design from the human mind of where we want to end up, and then the best chemistry that we can that's automated. And so I said that this title has popped into my head. This is a ChemoGrid, but the variation of this is for a nice popular name would be Genesis engines, an engine designed to get to a biogenesis or a second abiogenesis on the Earth. And I started putting out the word to people who've been involved with the EvoGrid, and then neighbors, friends and neighbors, yourself. And we have about 18 authors recruit. I still need another 10 authors.
Gosh.
So, and 6 chapters have come in. The early deadline was the 31st of July. 6 chapters have come in. Dave's draft chapter came in this morning. And it's fantastic work. So what we've got, in order to figure out this highly multidisciplinary approach to origin of life, we've got complexity theorists. We've got Stuart Kaufman writing a chapter on how he thinks novelty occurs in the universe. Another method that is relevant to building machines like this. We've got biochemists, good biochemists writing on this. Experimentalists who have approaches of how to build chemogrids, people who do microfluidics for a living, and robotically controlled chemistry. We have a philosopher who's written about the moral status of what comes out of these machines, a public policy person, and a wonderful writer, Robert Pollack from Columbia, who's already contributed the first of 2, what I'm hoping will be 2 prefaces. And it's basically, why do we do this? Why are we trying this? Beautiful preface that he's already written.
So there's an interesting question associated with why we do this. And I think certainly, having come from the Artificial Life Conference, there was a group of folk who will remain unnamed who had gone through the original protocell circumstance, particularly associated with startups and these kind of things. And I did have an opportunity to meet Steen at the conference, which was wonderful as well. He's kind of carrying the flag on through the choppy waters of artificial life specifically. But there's certainly a group, I don't know how I would characterize it, that have gone through the early protocell stuff, that see this very, very long term and kind of slow iterations at best. How do you avoid this being something that becomes like virtual reality, or as we've discussed previously, avatars, where there's a huge upspike thanks to folks such as Dima and others, and then, the buoyancy of actual deliverables is not necessarily seen, and then there's a downward progression. I mean, we've talked about this associated with the winters that come through these things. What do you see will buoy on this origin of life, this Genesis engine, through potentially initially choppy waters.
This is because, and I would say the groups that are failing are the groups that really don't know the code they're writing, which is it's chemistry code. And I can give you and they're not using the latest languages and techniques. So, not trying to toot Dave Deemer's horn, but what they're doing, Dave Deemer, the techniques that they're using are so cutting edge, and they're so code chemistry. Here's one. Late 1980s, Dave was in his lab, and they notice that a piece of DNA next to a membrane has somehow gotten itself through a micropore in the membrane, but it's gone all the way through, and you're talking, millions of base pairs have gone through. It was on this side of the membrane, now it's on that side of the membrane. Write that up and say this is pretty unusual. No one's ever seen this in the lab. How did this mechanism work? Well, there were 2 or 3, 4 citations in the first few years. Now there's like 25,000 citations. A whole company was formed called Oxford Nanopore. Millions of investment, because it turns out this is a discovery that's profound. You can do individual molecular counting using a micropore through a lipid bilayer, a specially designed micropore where literally things are moving and they're clicking physically as it's measuring. So in April, there was a conference in Florida that Dave was at, because of course Dave's on the board of this company, everything advisor, and a guy held a box up and he said, "This will sequence the human genome in hours when we launch this thing." So it's a technique that is profoundly important. There's another group doing X-ray technique, direct X-ray on 2D structures, and being able to measure the chemical contents without dyes, without doing mass spec and destroying the sample. They can actually just basically bounce X-rays off this stuff and tell you exactly what's there. So you put all this together, the individual counting, the wet drying cycles, the automation through well plate technology. This is the kind of oomph you need to do this kind of work. You need all the latest techniques, the best languages, the best thinkers. And if you don't have those, you can't write the code.
So there's an interesting, and this is part of my chapter that I'm contributing to the book, there's an interesting open source analogy here. The companies, and certainly this was my observation, and clearly there's historical legacy and human emotion invested in this as well. But the companies that have been able to look out and been able to say, this component is something that is a breakaway, and the researchers as well, this is the example of Dima, but let this go off and live in its own, versus the completely closed companies. It's an interesting metaphor that basically, this is part of my chapter associated with what artificial life can teach this emerging aspects of engineering and aspects of open source. But what you're describing is fundamentally open source.
It's fundamentally open source, and there's a huge economic driver behind this.
Certainly.
So it's like the game industry drove 3D chipsets, which led to the boom of a lot of other things. So instead of operating in a vacuum, doing your artificial life work or doing your origin of life work as a side thing, which a lot of Nobelists do. They have an origin of life because they have their reputation already, and they can do these funny little things that don't have much commercial value. What Dave has done, and what the Genesis engines thing is, you can go to a pharma, and you can say, Genesis engines are designed to do the search through chemical space to a very complex pre-life structure that it has behaviors. Can you imagine what you could do with this to do drug design?
So my hypothesis that I put out there, particularly from the early interaction with a fellow from Lilly in the Biota Live, actually, is that probably, and I know you have some ins into pharmaceutical companies as well, there are these early elements that are existing, maybe more advanced than we know, but because of the closed nature of the way that this is done, the lasing that you get in open source, the rich interaction, is lacking in these areas. If these people can be coaxed out, if this work can be coaxed out, I think it would be very interesting and clearly contributive.
And the book will do that, because in the commercial application section that somehow gets written, I mean, if you can build a piece of machinery that can guide a complexification vector through chemical space, you can produce any molecular structure. You can set a teleological end goal of saying we need something that has this chemical structure because we know it can act this way on a virus, or it can move through this kind of a cellular membrane, and it's a lock and key. But we don't know the pathway by which to build the industrial chemistry to make this thing. We have it. We see it in nature. We can. So a Genesis engine can show you pathways to manufacture chemical things in efficient ways, higher and higher efficient ways, because you basically are going to say, okay, that was a 1,500 step process, and we've got the molecule. We don't have it in big enough yield.
And then you can work on optimizing those 1,500 steps down.
Optimize those steps,
yes.
and it's automated.
Yes.
And so now you can design not only the molecule, can design the manufacturing process to be optimal and higher yields. And so you can take the same, you can piggyback on that kind of thing. When the pharma industry and the other industries see this is the approach, you'll get all the funding, and you'll get the spin-off of being able to use generative computing systems to use it in origin of life work. And so it's just a matter of putting that whole thing together, getting program managers involved.
Mm-hmm.
We're talking NIH, going to Glaxo, running it past. I have friends at Glaxo who are senior researchers. And do a talk there and say, this is the approach. These systems done professionally are going to, they're going to be $10 million systems, and seeing if investment, this is a revolution in what we do.
And they probably have some of the pieces there already.
They probably have,
and that's the interesting thing.
Yeah. The Human Genome Project is part of
certainly
of this.
Yeah, is the barriers for entry are probably relatively small. So let's talk about the book as a thing. In terms of your need for an additional, I know Setback in particular has a legacy of these super books. But do you see this more potentially in a series, and in terms of getting the additional 10 authors with the view that you already have stuff coming in, do you think it can be split up into a smaller book, or is the requirement that it has to have 25 plus authors?
I think it always, for that series, we're locked into that. And in a sense, this is my out-of-town tryout. This is getting a book about this out, but as it matures, a standalone book that's not part of a series would come out. So this is the book that's the fishing expedition of who has something to say, what are the basic themes. It's fairly easy. I mean, it's not a book that you have to sell a certain number of. It's not an MIT Press book.
Yeah.
It's not a... So this is the putting it together for the first time.
And I've been involved with half a dozen or so of these Setback Gordon et al. Books, Swan, etc. I guess my broader interest in them is I really love thumbing through them once they come out. You need to form a community which is secondary to these books, unfortunately. And I guess my frustration, although I have gotten some correspondence from writing in previous of these Setback Springer books, is that there isn't more into collaboration. How do you think your book might be different in terms of actually motivating the authors into collaborating?
What you do at that point, so this is all aimed at the ISIL meeting in Japan in 2014. So in Nara, Japan, they're having International Society of Origin of Life next conference, which is like the Origins was in Montpellier. They're only every 3 years. So what I'm aiming at, and Dave is, involved with ISIL, is a number of these authors who enjoy writing for the book, think it's innovative, it's revolutionary, innovative. Let's have a panel. Let's start this thing out as a community. Let's do a panel and discuss or a track on doing ChemoGrid Genesis, which is automating the quest for the origin of life, and get that done at that conference. People recognize each other. Dave shows his system for master biology. There's somebody else who shows their system, and they realize in their heads we're dealing with a common approach here. Do we have our own symposium? Do we, submit more papers together and call it such and such, because we're going to do it anyway. And I've done this 3-4 times before. I did it with avatars and
certainly without question. Biota, yes.
Biota. It was all finding people who already were working on something. They identify that, yes, you're working on that too, and here's a name for it. And
so this area, the area that we live in, has a small degree of luxury, and we're soon to welcome Larry Yeager back into the area. In terms of folk who are writing in the book who also live in this area, I mean, there are 3 of us that you've mentioned so far. Are there more?
I need to actually pluck a few more from this area. I have 2 or 3 more authors on dock to try to get. I went to see Freeman, and he's really interested in the book, but he doesn't want to commit to any.
No, I think yes, time. That is yes.
But I'm hoping that he would potentially review the book, which would be wonderful.
So in terms of just the local community, it would be wonderful, and I think you're probably the equidistant location for the 3 of us. It would be wonderful to be able to get together, maybe record some audio or just have a casual conversation, maybe even meet in Santa Cruz. Because certainly my sense of this book is very much in isolation, but I'm certainly absolutely fascinated by the stuff that the Demon and others are doing in this area, and I think the idea of collaboration should be more than just a book framing. I think basically, particularly in terms of, as you've described, Dima's insights in engineering fundamentally in this new field is certainly something that I'm very receptive to, and I'm writing on in my chapter as well.
In fact, one opportunity is the NASA Astrobiology Center meetings, which are about every couple of months, and those have great presentations. Andrew Pohoreal, anybody working in astrobiology, and they're in Origin of Life. So that's one place. It's sort of like the gray thumb of Origin. But you're absolutely right. If there was some kind of... And the DIY bio community, I put out a call on their list, do-it-yourself bio. There's John Cumbers over at Ames of the BioBricks people.
Yeah, I would certainly consider him as one of the people who would be ideally suited if we could get a small group together to have a jamming session.
And it's almost like if we attached it to the DIY bio meetings, the meetups that they're having already, and have Dave come and do a presentation at one of them, and just say, can we form a DIY, Origin of Life, DIY bio / Origin of Life / ChemoGrid, and get some of the thinking started. I've been just overwhelmed.
Certainly.
But as usual.
So for the benefits of folks listening in, my wife and I had the amazing opportunity of going through some of Bruce's archives. I'd seen the Digibone previously, but this kind of edge of Biota, but the Timothy Leary archive. This is a guy who had collected together... He had artificial life books that I had never seen previously, including a Japanese one that I'm going to have to track down. Clearly, and it was a beautiful full circle, because I'd heard previously, I think through you and potentially others, that Timothy Leary did have some early interests in artificial life, and I've seen that represented now in his library. But also we talked a little bit about my time with Roy Plotnick as another person, completely different field, but who reaches out and has picked up a good portion of the artificial life community as well. But in terms of what you'd like to see from the artificial life community, with the view that there are so many folk that are in so many interesting areas coming together, potentially contributing to this book, potentially just being a sounding board.
Yeah.
What's your sense of the community and how it can integrate into this?
I think that my plea for the community is think outside of the box, and literally outside of the computer box. Because if you start to study how life works and how complex it is, and the vast size of machinery that it is, and you realize that everything we're doing in artificial life in silicon is a toy. It's a complete toy. It's such a toy that it may not really have any relevance to
Well, that's
biology.
I think that's an interesting point, because even within toys, you can create microcosms and when you look at what biologists do, particularly a potential aversion to complexity in a lot of their experimentation, a lot of what the hard science is in this area as well could also be described as being a toy in terms of simplification.
They try to simplify.
Exactly.
Yeah.
So it is a difficult inclusion in some regard, but I think my sense of coming from the Artificial Life Conference and spending time with academics is that we all need to embrace complexity in whatever form. An area that you haven't touched on, but something that I think is really critical here and potentially resonates, is the hard artificial life community. Not only hobbyist robotics and these kind of elements, but you're describing things like Arduinos, things that basically the hard artificial life / robotics / hobbyist robotics community is already a part of. Clearly they can contribute a lot here as well.
They can.
Can you talk a little bit on that?
I think the grand unification of all of this is saying, let's take a non-trivial, high-performance computing environment called chemicals. Let's take the best robotics, the best hard artificial life that we've got, which is high-performance, parallel system modeling, multi-core modeling, vision systems, edge detection, whatever, the best robotics, and combine them with the best design, the best human design of complex experiments, of complexifying experiments, and create a single universe, a single system that can deliver. Because then in each component is doing a job appropriate to its abilities. The human mind's designing the overall thing. The chemicals are the non-trivial. They're nature. You're literally computing with nature. They do nature. They've always done nature. They can do it in real time.
Yes.
And then the best vision, the computers are really good at search and selection. They're lousy at simulation. They're great at search and selection. They're lousy at parallelism and all this non-determinism.
Well, historically, at least.
Historically. Well, one of the things I did in part of the EvoGrid for the PhD is I went to the Institute for Advanced Study, and they brought out all the archives. They brought out von Neumann's. They brought out Oppenheimer's director's files for the ECP, the Electronic Computer Project, which is John von Neumann's computer at Princeton at the Institute. And I went through everything and discovered all these letters about, we're building a contingent architecture. We're building something just so that we don't have to set up patch cords for 30 days to run a 15-minute calculation. We're building something that will be reliable and will work, but it's not the end-all and be-all architecture. It's a contingent, and of course it is the architecture we're still with.
To a certain extent. I mean, I think what has happened in terms of multi-core processors and a lot of the optimization that's gone through that has changed some of that groundwork.
But it's still a primary and secondary cache, a stream of instructions being, whether or not it's a pipeline or not.
Well, true. It's certainly its lineage is well noted.
It's well noted. So the thing was, my contention was that computers are so far separated from how nature works that we can't shoehorn nature into them. Therefore, we have to marry them with nature.
But we don't know how nature works fundamentally. We can make assertions about how nature...
But we know that it's non-deterministic, it's stochastic, it's mostly dissipative, mostly nothing happens most of the time. So there's a huge amount. It's and we know how entropy works.
Certainly.
So, and all of those things are super hard to simulate in silicon. They're super hard to do. And because people get bored because nothing's happening for the longest time. But marry nature. This is why I think robotics are such a huge area for learning, because it's real nature married with computing systems that are parallel models. At NASA for years, we did the simulation of robots, of rovers going down slopes, craters on the moon, and things like that. And initially, it was just rigid body dynamics, and then it became more soil compression dynamics. But it took 10 years to get to the point where we could actually get a realistic movement and slippage of virtual wheels, kind of like Curiosity will do when it's going down a slope. And it took 10 years to get there. And it struck me at that time that build a physical robot out of metal, put it in a sandbox, wire it up with a zillion sensors, and have a parallel virtual model going.
Certainly.
And then your parallel virtual rover can calibrate with the real one based on nature. And you can develop really rocking, incredibly effective models that you could use to drive on the moon. So that was the genesis of the idea of doing this. Of always have nature in your system as your primary processor. Your primary core is nature. And then cluster, robotics and computing and artificial life models and GAs around it, and you will create something absolutely new that can really go to frontiers. It's not going to be self-limiting because you're trying to keep up with the chemistry, or you're trying to keep up with the movement of a wheel through, because that's the richest computing environment, and you're trying to harness that computing environment and work with it, and it will pull you a lot farther, a lot faster. And Mark Bedaux's whole 10 points about the future of artificial life, he points this out, that you need to do hybrid systems, that you will learn so much more. It's like the Grand Challenge, the DARPA Grand Challenge. They did that because they realized robotics is going nowhere in 30 years. Let's put these cars on rough track roads, with no one else driving on them, and robotics move, and vision systems and edge detection systems and hazard avoidance systems evolve so fast in the 2 or 3 grand challenges, because the embarrassment of running a 3-ton Hummer off the highway and hitting a bridge abutment. Those teams actually got down to work because they had to deal with nature.
Certainly.
So I think this is an opportunity for the artificial life community to grapple with real nature.
So this is science and engineering, fundamentally.
Science and engineering.
Yes. And
then you're not only, it creates relevance for the artificial life community, because if you're a complexity scientist, you may be not have a lot of work, because that has sort of come and gone, the complexity theory and that kind of thing. But if you're a complexity scientist who can build things that can do broadband, long procedure, chemical experiment pathways, because you work on the system, they'll have suitors lining up at your front door, and funding, because you're now dealing with nature. You're now using your skills to help a natural process. And in the case of they talk about drug discovery. This isn't just drug design. I mean, if you can build systems like it's not just drug design, it's the design of the industrial process to make the drugs, and that's the step that's the hardest on the manufacturing.
Certainly.
And and if we solve the origin of life along the way, then it's a triumph. It would be a triumphal merger of the artificial life community and the theoreticians like Kaufman and the great bench chemist workers like a Dave Deemer, all working together. Would be the greatest triumph, that you not only build a system that is the most complex hybrid computing natural system ever built, but not only it can show you how the pathway to life was, but it can also it will revolutionize all manufacturing in the 21st century.
Well, Bruce, you've given the vision once again. Now many of us have to scurry away and start the work, but it's always a pleasure chatting with you, and I look forward to talking with you maybe when the book's closer to coming out, and potentially other stops along the way. So thank you very much for the chance to chat with you.
It's always a pleasure.